Friends, hello students. Today we will learn about protoplast culture and somatic hybridization. Protoplasts are naked plant cells without the cell wall, but they possess plasma membrane and all other cellular components. They represent the functional plant cells, but for the lack of the barrier, that is cell wall. Protoplasts of different species can be fused to generate a hybrid, and this process is referred to as somatic hybridization. Let's know about another term, hybridization. What do you mean by hybridization? Hybridization is the phenomenon of fusion of a normal protoplast with an enucleated, means without nucleus protoplast that results in the formation of the hybrid or cytoplast. Now let's know about the importance of protoplast and their culture. The isolation, culture and fusion of protoplast is a fascinating field in plant research. Protoplast isolation and their cultures provide millions of single cells for a variety of studies. Protoplasts have a wide range of applications. Let's know about the applications. First is the protoplast in culture can be regenerated into a whole plant. Second is hybrids can be developed from protoplast fusion. It is easy to perform single cell cloning with protoplast. Next point is genetic transformation can be achieved through genetic engineering of protoplast DNA. Protoplasts are excellent material for the on-structural studies. Isolation of cell organelles and chromosomes is easy from protoplast. Protoplasts are useful for membrane studies. Next is isolation of mutant from protoplast cultures is easy. Now let's know about the isolation of protoplast. How do we isolate protoplast? Protoplasts can be identified in two methods. One is mechanical method, another is enzymatic method. Let's know about the mechanical method. Protoplast isolation by mechanical method is a crude and tedious procedure. This results in the isolation of a very small number of protoplasts. First point is a small piece of epidermis from a plant is selected. Second is next point, the cells are subjected to plasmolysis. This causes protoplasts to shrink away from the cell walls. The tissue is dissected to release the protoplast. Mechanical method for protoplast isolation is no more in use because there are some limitations. First limitation is its yield of protoplast and their visibility is low. Next is it is restricted to certain tissues with vacuolated cells. Next is the method is laborious and tedious. However, some workers prefer mechanical methods if the cell wall degrading enzymes cause deleterious effects to protoplast. Now let's know about the second method, that is enzymatic method. Enzymatic method is a very widely used technique for the isolation of protoplast. The advantage of enzymatic method include good yield of viability cells and minimal or no damage to the protoplast. Now let's know about the sources of protoplast. Protoplast can be isolated from a wide variety of tissues and organs that include leaves, roots, shoot apex, fruits, embryos and microspores. Among these, the mesophyll tissue of fully expanded leaves of young plants or new suits are most frequently used. In addition, callus and suspension cultures also serve as good sources for protoplast isolation. Now let's know about enzymes used for protoplast isolation. The enzymes that can digest the cell wall are required for protoplast isolation. Chemically, the plant cell wall is mainly composed of cellulose, hemicellulose and pectin, which can be respectively degraded by the enzyme cellulase, hemicellulose and pectinase. In fact, the various enzymes for protoplast isolation are commonly available, commercially available. The enzymes are usually used at the pH of 4.5 to 6.0. Temperature should be 25 to 30 degrees centigrade with a wide variation in incubation period that may range from half an hour to 20 hours. The enzymatic isolation of protoplast can be carried out by two approaches. First is two-step or sequential method. Second is one-step or simultaneous method. Let's know about first is two-step method or sequential method. The tissue is first treated with pectinase to separate cells by degrading middle lamella. These free cells are then exposed to cellulase to release protoplast. Pectinase breaks up the cell aggregates into individual cells, while cellulase removes the cell wall proper. Now, second method, one-step or simultaneous method. This is the preferred method for protoplast isolation. It involves the simultaneous use of both the enzymes and macroenzyme, microenzyme and cellulase. Let's know about the isolation of protoplast from leaves. Leaves are most commonly used for the protoplast isolation since it is possible to isolate uniform cells in large numbers. The procedure broadly involves sterilization of leaves, removal of epidermal cell layer, treatment with enzymes, isolation of protoplast. Let's know about the purification of protoplast. How can we purify the protoplast? The enzyme digested plant cells besides protoplast contain undigested cells, broken protoplast and undigested tissues. The cell clumps and undigested tissues can be removed by filtration. This is followed by centrifugation and washing of the protoplast. After centrifugation, the protoplast are recovered from charcoal. Now let's know about the viability of protoplast. It is essential to ensure that the isolated protoplast are healthy and visible so that they are capable of undergoing sustained cell divisions and regenerations. There are several methods to assess the protoplast viability. Fluorescence diacetyl staining method, FDA method. The dye accumulates inside viable protoplast which can be detected by fluorescence microscopy. Next is phenolphthalein stain. Phenolphthalein stain is selectively taken off by dead protoplast while the viable cells remain unstained. Exclusion of Ivan blue dye by intact membranes. Next is measurement of cell wall formation. Chalcofluor white CFW stain binds to the newly formed cell walls which emit fluorescence. Oxygen uptake by protoplast can be measured by oxygen electrode. Next is photosynthetic activity of protoplast. Next point is the ability of protoplast to undergo continuous mitotic division. Now let's know about the culture of protoplast. how we can culture the protoplast. The first step in protoplast culture is the development of a cell wall around the membrane of the protoplast. This is followed by the cell wall division that give rise to a small colony. With suitable manipulation of nutritional and physiological conditions, the cell colonies may be grown continuously as cultures or regenerated to whole plants. Protoplasts are cultured either in semi-solid, agar or liquid medium. Sometimes protoplasts are first allowed to develop cell wall in liquid medium and then transferred to a agar medium. Now let's know about the agar culture. Agarose is the most frequently used agar to solidify the culture media. The concentration of the agar should be such that it forms a soft agar gel when mixed with the protoplast suspension. The plating of protoplast is carried out by Bergman's cell plate technique. In agar cultures, the protoplast remain in a fixed position, divide and form cell clones. The advantage with agar culture is that clumping of protoplast is avoided. Now let's know about the liquid culture. Liquid culture is the preferred method for protoplast cultivation for some reasons. Let's know about the reasons. First is it is easy to dilute and transfer. Second point is density of the cells can be manipulated as desired. Next point is some plant species the cells cannot divide in agar medium, therefore liquid medium is the only choice. Next point is osmotic pressure of liquid medium can be altered as desired. Now let's know about the culture media. Nutritional components. What nutrients should a plant need? In general, the nutritional requirements of protoplast are similar to those of the cultured plant cells, callus and suspension cultures. Mostly MS and B5 media with suitable modifications are used. Let's know about some features. The medium should be devoid of ammonium, and quantities of iron and zinc should be less. The concentration of calcium should be 2 to 4 times higher than used for cell cultures. This is needed for membrane stability. Next point is high oxygen or kinetin ratio is suitable to induce cell divisions, while high kinetin or oxygen ratio is required for regeneration. Next point is glucose is the preferred carbon source by protoplast, although a combination of sugars can be used. The vitamins used for protoplast cultures are the same as used in standard tissue culture media. Now let's know about the osmotic pump and osmotic pressure. What is the difference between these two terms? Osmotic pump broadly referred to as the reagents or chemicals that are added to increase the osmotic pressure of a liquid. The isolation and culture of protoplast require osmotic protection until they develop a strong cell wall. In fact, if the freshly isolated protoplasts are directly added to the normal culture medium, they will burst. Thus, addition of an osmoticum is essential for both isolation and culture medium of protoplast to prevent their rupture. The osmotica are of two types, non-ionic and ionic. Non-ionic osmotica. Let's know about the non-ionic osmotica. The non-ionic substances most commonly used are soluble carbohydrates such as mannitol, sorbitol, glucose, fructose, galactose and sucrose. Mannitol, being metabolically inert, is most frequently used. Ionic osmotica. Potassium chloride, calcium chloride and magnesium phosphate are the ionic substances in use to maintain osmotic pressure. When the protoplasts are transferred to a culture medium, the use of metabolically active osmotic stabilizer, that is example glucose and sucrose, along with metabolically inert osmotic stabilizers, is advantageous. As the growth of protoplast and cell wall regeneration occurs, the metabolically active compounds are utilized, and this results in the reduced osmotic pressure so that proper osmolarity is maintained. Now let's know about the culture method. The culture techniques of protoplast are almost the same that are used for cell culture with suitable modifications. First is feeder layer techniques. Feeder layer technique. Let's know about feeder layer technique. For culture of protoplast at low density, feeder layer technique is preferred. This method is also important for selection of specific mutant or hybrid cells on plates. The technique consists of exposing protoplast cell suspensions to X-rays and then plating them on agar plates. Now let's know about co-culture of protoplast. Protoplast of two different plant species can be co-cultured. This type of culture is advantageous since the growing species provide the growth factors and other chemicals which helps in the generation of cell wall and cell division. The co-culture method is generally used if the two types of protoplast are morphologically distinct. Now let's know about microdrop culture. Specially designed dishes, namely Q-Park dishes with outer and inner chambers, are used for microdrop culture. The inner chamber carries several wells wherein the individual protoplast in droplets of nutrient medium can be added. The outer chamber is filled with water to maintain humidity. This method allows the culture of fewer protoplast per droplet of the medium. Now let's know about the regeneration of protoplast. Protoplast generation, which may also be regarded as protoplast development, occurs in two stages. First is formation of cell wall. Second is development of callus or whole plant. First, let's know about the formation of cell wall. The process of cell wall formation in cultured protoplast starts within a few hours after isolation, that may take 2 to several days under suitable conditions. As the cell wall development occurs, the protoplast lose their characteristic spherical shape. The newly developed cell wall by protoplast can be identified by using chalcofluor with fluorescent stain. The freshly formed cell wall is composed of loosely bound microfields which get organized to form a typical cell wall. This process of cell wall development requires continuous supply of nutrients, particularly a readily metabolized carbon source. Cell wall development is found to be improper in the presence of ionic osmotic stabilizers in the medium. The protoplast with proper cell wall development undergo normal cell division. On the other hand, protoplast with poorly regenerated cell walls show budding and fail to undergo normal mitosis. Now let's know about the development of callus or whole plant. As the cell wall formation around protoplast is complete, the cells increase in size and the first division generally occurs within 2 to 7 days. Subsequent divisions result in small colonies, and by the end of third week, visible colonies are formed. These colonies are then transferred to an osmotic free medium for further development to form callus. With induction and appropriate manipulations, the callus can undergo organogenic or embryogenic differentiation to finally form the whole plant. The general view of the protoplast isolation, culture and regeneration is is isolated. Plant regeneration can be done from the callus obtained either from protoplast or from the culture of plant organs. There are, however, certain differences in these two caluses. The callus derived from plant organs carries performed buds or organized structures, while the callus from the protoplast culture does not have such structures. The first success of regeneration of plants from protoplast cultures of Nicotiana tabacum was achieved by Tech B. et al. in 1971. Since then, several species of plants have been regenerated by using protoplast. Now, let's know about the subprotoplast. What do you mean by subprotoplast? The fragments derived from protoplast that do not contain all the content of plant cells are referred to as subprotoplast. It is possible to experimentally induce fragmentation of protoplast to form subprotoplast. This can be done by application of different centrifugal processes created by discontinuous gradients during centrifugation. Exposure of protoplast to cytochalcin B in association with centrifugation is better approach for fragmentation of protoplast. There are three types of subprotoplast. First is mini protoplast, cytoprotoplast and microprotoplast. Let's know the mini protoplast. Mini protoplasts are also called as karyoplasts and contain the nucleus. Mini protoplasts can divide and are capable of regeneration into plants. Next is cytoplast. Cytoplasts are subprotoplasts containing the original cytoplasmic material but lack nucleus. Thus, cytoplasts are nuclear-free subprotoplasts which cannot divide but they can be used for hybridization. Microprotoplast. Third point is microprotoplast. This microprotoplast was suggested for subprotoplasts that contain not all but a few chromosomes. Now let's know about the somatic hybridization. What do you mean by somatic hybridization? The conventional method to improve the characteristic of cultivated plants for years has been sexual hybridization. The measure in limitations of sexual hybridization is that it can be performed within a plant species or very closely related species. This restricts the improvements that can be done in plants. The species barriers for plant improvement encountered in sexual hybridization can be overcome by somatic cell fusion that can form a viable hybridization. Thank you. Next discussion we will discuss in second part. Thank you.